If you look at Darwin's tree and you look at the base of the tree and focus there, it turns out that there's a critical problem that all evolutionary biologists today acknowledge has been unsolved by any undirected evolutionary mechanism. And that is the problem of the origin of life. Now, in Darwin's time, scientists didn't worry about this so much. They knew that that Darwin didn't propose a theory for how life began in the first place from simpler non-living chemicals. But they also assumed a view of life that suggested that life wasn't all that complicated, and therefore that it didn't really manifest much of even an appearance of design. The 19th century view of the cell was that it was a simple a little glob of jello. We knew from microscopes at the time that the cell was an enclosure and that there was some kind of chemistry going on in there, but scientists at the time assumed that it was very simple chemistry, that with a couple, they actually had a name for the substance they thought was the essence of life. They called it protoplasm. And so one of Darwin's contemporaries, Thomas Huxley, said the cell is a simple, homogeneous globule of protoplasm, or in another place he said, a simple homogeneous globule of undifferentiated protoplasm. It's just chemical jello. And if you think of life in that way, it's not hard to think about how life might have arisen in a one or two step process. You might have a few simple chemicals, they react to form some slightly more complex chemical compounds, they react, and boom, you've got the protoplasm. That was the kind of idea that people had about how life must have began in the 19th century. Now, I love this quote from Huxley because it makes me feel so smart. This is one of the greatest biologists of the 19th century, and he was completely ignorant of everything that we now know about the complexity, the integrated complexity of the inner workings of life. So let's look forward a little bit and look at some of the things that have been discovered that have completely altered our view of life and completely and made more difficult this problem of he said that at the heart of the problem of the origin of life lies a fundamental question, and that question is, what is it exactly that we're trying to explain the origin of? Awkward phrase, the origin of, but that's, what are we trying to explain the origin of? If you think life is simple, if it's just a simple chemical jello, it's pretty easy to imagine that you could produce life in a couple simple chemical steps. But if you think of life as something like a chemical factory run by digital code, which is what we're now beginning to realize, then it becomes much more difficult. And one of the first things that was discovered that began to break down this simplistic concept of life that made scientists think it was so easy to explain its origin was the discovery of the structure of proteins during the 1950s. And I like to use a more precise phrase to describe what was discovered about proteins. We discovered that they were very complex and also that they were very specific or specified. And I'll explain what I mean by that. Let's start with some pictures of proteins. Proteins, first of all, are the molecules that do all the important jobs inside cells. They catalyze reactions, they build structural parts, they process information on DNA, which we're going to talk about in a minute. So they do all the important jobs that keep living systems alive. And up until the 1950s, scientists had very simplistic ideas about proteins, just as previously they had very simplistic ideas about the cell. They thought of them as like, that they would end up being things like hat boxes stacked on each other, or cigars all stacked like, or Lincoln logs or something like that, that they would be highly regular, very simple structures. The big surprise in the 1950s is that they turned out to be highly intricate, three-dimensional shapes, and even more important than that was the discovery that the intricate three-dimensional shapes that proteins have is critical to their function. So I've got a slide here of a protein, an enzyme protein, the job of which is to break apart a two-part sugar. And you can see that the sugar, which looks on the slide like a little barbell, nestles into the shape of the protein that is going to catalyze that breakage. And you can see that there's a beautiful kind of hand-and-glove fit between the enzyme and the sugar that enables that chemical reaction to go forward. And that's the case with almost all proteins. They do their job because of a lock key or more aptly a kind of hand-in-glove fit, where one molecule matches spatially the other. And that three-dimensional specificity is the key to the function of the protein. But it turns out that that three-dimensional specificity derives from a more fundamental one-dimensional specificity, a specificity of arrangement. And I've got a visual aid that makes this clear. This is these are SnapLock blocks. My kids are now teenagers, but I stole these from them when they were about 2 or 4 years old, because it said on the box ages 2 to 4. They're little colored beads of different shapes. And what I'm using them to do is to illustrate what proteins are made of. Proteins are made of smaller molecules called amino acids. And there are 20 different amino acids that are part of proteins. And proteins only become proteins. They only acquire those intricate three-dimensional shapes I was talking about when the amino acids line up in a very specific sequence or order, and then as a result of their lining up properly, they exert forces on each other and cause the chain to fold into a specific